All right, I think we're going to go ahead and get started. So welcome everybody to, this is the last session of Nano at Tech for this semester, the spring semester 2022. So I don't have an announcement for the next speaker. That'll be in August, and of course you'll get the usual emails and see the posters and all of that. Before I introduce today's speaker, I do want to give some thanks to a couple of folks here in IEN who have really helped out with this seminar series over certainly this semester and even beyond. So Amy Duke, who handles all the communications with the speakers, Lori Hay, our communications and marketing manager, who prepares all of the marketing materials, and Mike Carson, who handles all of the room logistics and the food. So I want to thank the three of them, certainly. So it's a real pleasure to welcome today our speaker, Aniruddh Sarkar, and we were just talking before I started. Aniruddh started in 2020 here at Georgia Tech, so really nobody knows who he is. So this is kind of his coming out party. Aniruddh got his bachelor's and master's degrees in electrical engineering at the Indian Institute of Technology in Bombay before coming to the United States to get his PhD in electrical engineering and computer science with a minor in biology at MIT. And then he stayed in the Boston area as a research fellow at the Ragone Institute and had associated positions at Massachusetts General Hospital, MIT, and Harvard University before, as I said, coming to Georgia Tech just 2 years ago, where he's currently an assistant professor in the Coulter Department of Biomedical Engineering here at Georgia Tech and Emory. And with that, I will turn it over to you.
Thank you. I think it's a small group, so we can keep it somewhat interactive. My title is a mouthful, but we'll get into parts of it as we go on. Usually I like to start this talk by showing this. So how many here recognize what this is? Okay, any more? I think maybe this is bifurcating by age a little bit here. Yeah, so this is, for the Star Trek fans out there, this is supposed to be the medical tricorder, right? Which in the year 2369, I believe, your doctor is supposed to be able to point it at you and diagnose what's wrong with you already, right? Just in this handheld little device. But that is not the, our reality, unfortunately, as of 2022. Of course, medical diagnosis, as of now, takes a whole lot of people, supply chains, and then a whole lot of equipment, which, as you'll see, a lot of it is from 50 to 100 years old technology still being used. But all of that eventually adds to delays in diagnosis, and the complexity of the instrumentation and the expertise required to run it results in high healthcare costs eventually as well. Now, of course, in the context of COVID-19, we've all become, unfortunately, more familiar than we want with the diagnostics issues, especially the scaling of the diagnostic pipeline, those kinds of issues. So, this is the typical RT-PCR test. It starts with the swab, but then eventually has to end up in a PCR machine somewhere. And scaling this, as you might recall from now almost exactly 2 years back, right, that this had been initially the challenge in a big way. And in fact, it can be argued that this contributed to how uncontrolled the pandemic became, where there were no accurate diagnostics available in most parts of the world, right? And even here, it has been argued that if you recall, the initial PCR kit released by the CDC had issues, right, primer dimers or whatever with the PCR, and there were errors there and so on. So all of that contributed to actually what the pandemic became. So in our lab, the question we sort of ask, or the hypothesis, the broader one we ask is, can miniaturization, right? So by miniaturization, I mean this which a lot of you might be familiar with, this idea of making chips smaller and smaller and more capable at the same time as they're becoming cheaper and cheaper as well, right? So this idea of miniaturization, can it actually do in healthcare what it has done in computation and communication, right? So can it help deliver scalable and affordable, in this case, diagnostics to the 10 billion people world that we are heading towards? So why do we think so? Well, there's a couple reasons. Of course, the scalable manufacturing is a big aspect of it. But also there is this interesting match of length scales now happening in electronics and biology, right? So these chips that we can now make fairly easily and inexpensively actually are at the same length scales of the unit features as, say, cells and bacteria and even viruses and individual molecules, right? So that gives us some unique abilities to use these chips to sense and manipulate individual biological objects. But what's more, at these length scales, so the micro and nano length scales that I refer to in my title of the talk, at those length scales, there are other physical phenomena happening in terms of fluids and electric fields and other energy fields. When you confine them to these length scales, certain unique phenomena happen that give you further capabilities to sense and manipulate biological objects as well. So this is broadly what we do in the lab, micro and nanoscale tools all across discovery, diagnosis, and therapy, so all aspects of healthcare actually, and I'll invite you to check out our website to see more details there. But for today's talk, I'm going to focus on two specific stories. One is biomarker discovery, which goes into what to measure. So a biomarker, marker of disease, what do you want to measure? Do you know what should be measured, right? And in that context, I'll talk about microscale immune monitoring, and I'll describe what that means later. But the advantages it brings is you end up using very low sample volumes, so diagnosis from a single drop of sample and so on. But also you're able to do large numbers of measurements at the same time, so high throughput, and large numbers of samples as well, so screening large numbers of patients, for example. All that feeds into discovering biomarkers. And then the other aspect I'll talk about is point-of-care diagnostics, which basically is taking these biomarkers, translating them to the clinic, being able to measure it right where the patient is in an inexpensive manner, right? And in that, I'll talk about this new transduction principle that we have developed in the lab using nanomaterials, integration of biomaterials and nanomaterials, which feeds into the electronic detection aspect of it, which then leads to being able to use these microfabrication techniques that I mentioned from the IC or microchip context, which leads to low cost, portability, and ease of use in diagnostics. So let's jump right into it. So I'll start by sort of reorienting ourselves, or bringing all of us to the same page. I mean, I know a lot of this is now familiar to everybody, but just to reiterate it here. So COVID-19, one of the major challenges from the diagnosis perspective is, right, that a lot of people get this viral infection and don't have even any symptoms. They can still be transmitting it, but they don't have any symptoms. Then there are people who have mild symptoms. And then there is a certain set who go on to have much severe symptoms as well. So this sort of heterogeneity of the disease is a huge challenge to diagnosis. It's not like you can say, hey, you're infected. So for example, in HIV, let's say, right, another virus, someone's infected, there's a very, very high chance that they will develop AIDS unless treated. So that's it. If they're infected, you only need to know that they're infected, and rest of your therapy or whatever you want to do, healthcare, is based on that. That is not so at all with COVID-19, and that's a huge issue. So what you want to do is, ideally, that early enough, maybe even when they're asymptomatic, you want an early diagnosis, but at that level, a huge fraction of the population is being infected. So we want it to be really scalable and cheap if you want to have an early diagnosis. But then the other more important even aspect here is, can you have some way of predicting beyond the known sort of correlations that we are all aware of, age, comorbidities, and so on? Can you have a more predictive or a prognostic marker that will help you to then, how you are apportioning your healthcare resources, who is getting more help, and so on, all those kinds of things, and then eventually helping individuals as well. So again, just describing the various infectious disease diagnostics modalities here, and excuse me if this gets boring. Like, how many people here have never taken an immunology class, for example? Right, so there's a significant number of us. I belong to that category as well, so I'll just assume and describe the basics with that assumption here. So in most infectious disease diagnostics, you are either looking for the bug itself, right, the thing that's infecting, the pathogen, or you're looking for the host response, which usually is the antibodies, but sometimes T cells as well, right? So what infects you can try to detect it, or you can try to detect the human body's response. One, as you might imagine, is a lot amplified, so easier to detect, right? The immune system generates a lot of response. The other may be a little rarer, but can still be detected depending on how large the infection burden is, for example. So in the context of COVID-19, of course, we know that RT-PCR, which basically detects viral RNA, so you're detecting the virus, is the reference standard, right? On the other hand, antibody detection, it's really not useful up until things like serosurveys. So they tell you not whether you are currently infected or not. They are at the population scale, epidemiologic scale. You're trying to find, okay, how much fraction of my population has been infected already, right? So it doesn't have much diagnostic use currently, at least. And that's because of this, right? Antibodies take a while to come on board. This is the IgG, the most common antibody used in serosurveys. That, in fact, can take up to a month even, but 14 days, 20 days, that sort of thing. IgM response can come up a little bit early. This is a different kind of antibody. But again, that's variable in people as well. So it's not really used for diagnostics much. It so happens that if you look at the engineering of this, right? So how do these diagnostics work? What instrument is needed? How to scale these diagnostics? The one that works for diagnosis currently is a high complexity assay. So this is where you'll see the pooling assays and things that, for example, Georgia Tech is doing. That's to put that central lab resource, which is expensive, in a central lab, but then pool a lot of samples and somehow be able to then you divide by those number of people and so on, and you're bringing the cost down, et cetera. So scaling to the individual diagnosis because of this complexity is tough for PCR. Versus antibody responses, relatively easier. They can still be challenging depending on what exactly you're trying to measure, quantitative or not, and so on. But they're moderate to low complexity, I would say. In terms of the predictive nature, right? We said the heterogeneity of the disease is an issue. You want to be able to predict. None of these work, basically. The viral load has been shown to be not predictive of disease course. People can have the same amount of virus and go totally different directions. The antibodies, of course, are not predictive as of now of anything at all. So the scientific questions which drives most of the work I'll describe today is, can we have an assay that can have a simplicity of serology? So serology is just a term for serum-based measurements, basically antibody measurements. Can we have a diagnostic which has the simplicity of serology and yet do early diagnostics, which is not possible right now, right? Because antibodies come on board much later only. Predictive serological monitoring. Currently, you either have antibodies or you don't, and there is no disease course prediction possible from just having it or not, basically, usually. But can there be more precise markers which are serological, antibody-based, but can be predictive as well? And yet, and of course, finally, the goal is to be able to, for such a disease, the goal is to be able to scale it as much as possible worldwide. So we'd like it to be good, sensitive and specific, but also simple, so that it can be done by untrained personnel. Personnel cost is a huge portion of these costs one is talking about, but overall low technology cost as well. So then just digging further into it, this is just showing the course of disease I described to you. And it is known, of course, that the asymptomatic to severe transition is correlated with age and comorbidities. It's not a perfect correlation. After hospital admission, though, so that's the definition of severe I'm using here, it's pretty much not known beyond that stage. Who ends up surviving and who doesn't is very difficult to predict at that stage. So yeah, that's just saying that this unpredictable disease progression actually ends up contributing to patient mortality as well. What is known is it's not the virus that's causing the bifurcation at that stage. you've gotten enough virus burden that you've landed in the hospital. It's usually the immune system, the immune dysfunction that leads to, that plays a much more important role in the disease progression at that stage, basically. So what we thought is, monitoring the immune response itself may provide a predictive biomarker. And then, of course, linking back to the science, if we understand how this bifurcation is occurring, maybe there is something mechanistic we can come up with, maybe better vaccines, who knows, or therapeutics, and so on. So there's the science aspect to this as well. So, of course, we are not the first to think of doing immune monitoring in COVID-19. So all arms of the immune system that you can imagine or know of, right? Cellular, so T cells, B cells, or humoral, cytokines, antibodies, all of these, various people have been trying to look at these things, and there's some correlations known. For example, lymphocytes go down with severity, T cell activation goes up, and so on. We, focus on antibodies, and with antibodies, there's been this sort of question in the field: are they good or bad? Now, you may say, well, antibodies are good. What are you talking about? I mean, I get these vaccines, and I'm told that I'm supposed to develop these antibodies, so they must be good. Well, it's not so obvious. So they are a correlate of protection from vaccines, that's for sure. It's also known that people who are immunocompromised, for example, they either lack seroconversion, meaning development of antibodies, or they have a delayed seroconversion. So that is linked to severity. So all that is pointing towards, of course, antibodies are good still. But the other interesting thing is the concentration of antibodies in people goes up with severity. It is well known that the more severe disease you develop, the more antibodies you have. So that's sort of troubling. Are they always good? Are there types of antibodies that are not good? So what is going on there? Why, or is it just a causation correlation thing that you're fighting the disease more and you're developing more antibodies, but they're still good? But that's not clear a priori for sure. And then there are, in some other diseases, although in COVID-19 now there hasn't been direct evidence of this yet, there is this concept of antibody-dependent enhancement. It happens in things like dengue fever, where antibodies actually bind to the virus and help it infect some other cells, because antibodies interact with cells as well. So that's a huge concern, that could that be happening in people who sort of after having severe COVID-19 are not able to recover. So antibodies, just again a little bit of the immunology here. Antibodies, of course, direct a large number of functions. So they don't just bind the virus, right? They in fact direct a lot of the functions in the immune system as well. A lot of the other cellular immunity is triggered by the antibodies that are bound to whatever pathogen one is talking about. So what we started to think about is that, well, everybody has antibodies, but are antibodies functionally, meaning in these functions that they direct, are they different in survivors versus non-survivors? And maybe that is the difference where it ends up being some antibodies are good and some antibodies are bad, right? So antibodies, of course, have thousands of different features, and usually, though, in diagnosis, when you get an antibody test, they just measure the concentration. In fact, the simplest one is yes, no, and then there is concentration, right? How much you have, basically. That's called titer. So, all these other features that antibodies have pretty much are neglected in diagnostics. So drilling that into it a little bit, this is what an antibody looks like. This typical Y-shaped molecule. At the front end is called the FAB end. It binds to the antigen, the virus. The back end is the FC end. That's what interacts with the immune cells on the back end. So it acts as a beacon, right? Bind to the virus, let the other immune cells know, hey, here's a virus, come eat me up, or come destroy this, or whatever that particular cell does, right? So what is known is this back end, although it is called FC, the C stands for constant, it's not really constant. Calling it a constant is a misnomer. The protein sequence in this part gets tuned, which decides what kind of antibody it is. And then there is the sugar that gets attached, which is highly tunable as well. So subclass and glycosylation sort of end up generating a kind of a functional barcode to the antibody. So the front end is binding to the pathogen. The back end has a barcode which tells the immune system what exactly to do with this. Neglect this, this is nothing. Or definitely come immediately and destroy this, this is really bad, right? So these kinds of things are being tuned on the back end of the antibody. So how does one read then this functional barcode is where we came in with these new technology ideas. What one wants to do clearly is measure the FAB, the antigen binding, right? We want to measure the antibodies that are viral specific, and the FC, so the subtype, glycosylation, FCR binding, and all this, and we want to do it simultaneously for all antibodies. Of course, we need to be sample sparing. We're saying we'll measure all these properties, hundreds of properties for the barcode and so on. We can't be using like a liter of blood or something to get this, right? These are rare molecules. So the sample use should not scale with the number of features we want to measure is a key property of the assay we want to develop here. So that essentially leads to it has to be highly multiplexed. So each measurement should give me a large number of readouts rather than one readout like a concentration or something. So we then, in this came in with this idea that we should use barcoded microspheres, which can be used to develop multiplex assays. This is a commercialized assay. It uses basically these beads which have a dye barcode. So it's a ratio of two dyes, one red dye and one IR dye, which is impregnated into the bead. But then on the bead, you can coat a capture molecule. And then you pool all these beads, right? They're barcoded, and then the barcode corresponds to what capture molecule is on the bead. And then you pool all these different capture molecule coated beads together, throw it into the normal assay protocols you have. So each sample is getting exposed then to all of them. And all of the different things you have coated are binding the different antibodies. So all of the measurements are happening, the bindings are happening in parallel, basically. And so this, there are various commercial vendors that make this. So up to 500 different readouts is what is claimed is possible. And then once you do that binding, you bind a probe, a secondary probe on it, which has fluorescence. Then you use something called a flow cytometer, which is basically a laser and photomultiplier tube-based readout of bead by bead, right? So you've got these two classification lasers, which classify the ratio of the dye here to identify the barcode on the bead itself. And then you've got a readout of the how much was bound. So that's a third thing you're reading, basically. So you get these arrays of 3 numbers, and then from that you knew which barcoded bead had which thing, so you can back out that particular readout or whatever that particular probe corresponded to in your case. So how does one apply this to antibody profiling, especially in the context of COVID-19? So in COVID, we know that the virus has a large number of different proteins. The immune system sees some of them more, some of them less. It depends on amount of that protein in the virus, but also how it's presented, right? Because some antigens are exterior, right? Most of the vaccines we are getting generates antibody to the spike protein, which is a surface protein. But there are other proteins inside, right? Once the cells get infected, they can display these antigens, and the immune system can generate a response to those as well, and so on. So what we did is, picked sort of based on some curation of the literature when we started this, 10 antigens in which we have some of these surface or more immunodominant antigens, including the spike to which most of the vaccines are targeted. But then some of the other less studied antigens, which are usually internal antigens. Because we had this capability to do highly multiplex measurements, we were like, well, why not? Let's see what's the immune system doing with these antigens. And then the other thing that is known, and I'll point out here, is there are other coronaviruses that circulate in the human population. They cause common cold-like symptoms. They've existed for a while, some up to 100 years, some 50 years, and so on. So those, of course, the human population has exposure to. So we were curious, does the existing response to those other viruses, they are similar, of course, in protein sequences and so on a little bit. Do they matter at all, the response? So we threw in some of those antigens as well. And then on the back end, so that's for the FAB, right, what antigen it binds to. For the FC end, we had this whole array of the antibody properties I talked about, subtypes, glycosylation, FCR, and so on. So that's 17 odd probes. So you can see for each sample, we are generating 170 readouts, right? So for each patient sample, 170 numbers eventually. Yeah, so this is just laying that out. You have we had an ICU cohort, so basically very severe COVID-19. On day 1, a blood draw is done, right, before it's known what's happening in terms of their clinical outcome. And then with that, with these barcoded beads, we take a drop of serum, so it's very sample efficient, really a little drop of sample needed. We bind it with that. We have the probes, and I described the flow cytometry-based assay to you. Eventually, you generate high-dimensional data, right? These 170 readouts of antigens, probes, and the patients, basically. With that, as I'll show you, to try to understand these large data sets, you have to use machine learning type techniques. So that's what we do. And I'll describe to you, what the labels here mean and how this helps us discover some biomarkers here. Right, so I'll start by talking about the canonical. By canonical, I mean this is the ones that people study more. This is the one that are in existing antibody diagnostics, for example. And just to orient you to this radar plot, which I'll use a lot. So each of the sort of sectors is one probe, one readout, right? And then the colors, the red is non-survivor, blue is survivor. So you're seeing, for example, the non-survivors have high IgG versus the survivors have high IgA. So you're starting to see some differences here. Yeah, particular kind of antibodies rising in survivors versus non-survivors, for example. And then you can do this with other antigens. So this was the nucleocapsid antigen. The one, if you get an antibody test today, this is what will be in that test, the nucleocapsid antigen. It's the largest amount of antigen. It's the antigen that SARS-CoV-2 expresses in the largest amount, so it's used in antibody tests, commercial antibody tests. But some of the other antigens, spike RBD, these are the vaccine antigens, for example. And I won't go into describing each of these in detail because eventually the conclusion here was that yes, there are differences, right? This is a heat map of fold differences, but none of these individually are even statistically significant, right? So it's very difficult to draw a conclusion here from individual univariate readouts. So this is where the machine learning comes in. Again, in the interest of time, I won't go into the details of the lasso, which is a down selection technique, and then PLSDA, which lets us lay this out in terms of the latent variables eventually here. But the point being made is you can start distinguishing, you can start classifying. So a minimum signature of 3 variables in this case, out of the 170 that we measured, can robustly classify survivors versus non-survivors. So that's what we started to see. And this is, by the way, with just those 3 antigens, right, which I call the canonical. The rest are still there, coming later. We see that there is high IgA in survivors. So that's interesting. This is an antibody that goes in all your mucous membranes usually, right? The nasal passage, lungs. So that makes somewhat sense. And then we see that some of this glycosylation I talked about, which tunes the back end, that's changing as well in survivors. The non-canonical antigens, which pretty much no one had studied till now, we start seeing that there are differences in them. So you see that the red and the blue bars are different lengths in many cases. And again, in this case, again, this is that fold ratio heat map. You start seeing some, in fact, univariate measurements even are statistically significant. NSP13, very little studied molecule. It's now known what it does, but the immune response to it is hardly understood. But we start to see that the survivors and non-survivors are differentially targeting this internal viral antigen, in fact. And again, same kind of lasso PLSDA techniques to do the classification. Interestingly, the non-canonical response, these are internal viral antigens, right, is equally good at predicting outcome. So this is sort of a surprise to us that the external antigens, it's understood that, or it is hypothesized at least that, well, you probably are blocking the infection, and that's why the vaccine is targeted to it. What are the non-canonical antigen antibody responses doing? Not so clear. But we do see some common features. There's still high IgA, even against the non-canonical antigens, high galactosylation as well, and then this NSP13 thing I pointed out. So there's a high IgG3, which is a particular subtype of IgG. We see a higher. So this made us sort of wonder what might be going on here, and we started thinking, well, maybe some of these other antigens, they are known to be quite homologous to the corresponding antigens in the endemic human coronaviruses. Maybe that existing response is involved here in helping out survival versus non-survival, but it wasn't clear yet. Then came the biggest surprise of this study. So we said, if this is an existing response, let's put in some pre-pandemic samples we have, right? This is before SARS-CoV-2 even came on board. This is from mid-2000s or something, some biobank samples. So we threw them in. And if you've gotten anything of how these polar charts, radar charts work, you'll see that the green samples, which are the healthy, have high responses, higher than the people who are COVID infected, right? This is pre-pandemic samples. they have an existing antibody response to these antigens especially, right? Not to the canonical so much, although there is some, but to the non-canonical antigens, pre-pandemic healthy people have an antibody response. So this was a huge surprise to us. And this again started pushing us down the route of thinking, clearly the endemic coronavirus infections must be doing something here because they didn't have exposure to SARS-CoV-2. How come they have an antibody response to these antigens, right? What is even more interesting, digging even more into it, not only is there a response, there is a distinct response, right? So the pre-pandemic healthy, if you do a three-way classification, you can build a pretty good classifier. You still see higher IgA in the survivors. All those properties come over from what we had there. But interestingly, we see the healthies pre-pandemic have a higher IgM response. So the M is memory, right? Just to refresh your memory here. So basically, these are memory responses. And so high IgM responses against non-canonicals in the pre-pandemic healthy is something we are observing here. So then, this goes a little bit into the immunology of the mechanism here. Is it that they have these antibodies, and then upon infection, who is able to switch to the more functional IgA class survives? Those are the mechanistic questions that this ends up raising here. And then, directly measuring the endemic. So if you recall, I mentioned we threw in the endemic coronavirus antigens directly as well, right? Up until now, I've talked about canonical and non-canonical SARS-CoV-2 antigens. Now, these are the antigens from the endemic coronaviruses. We see that even the response to the endemic coronavirus antigens only. So if you don't measure the response to the SARS-CoV-2 antigens at all, you measure response against only the endemic coronaviruses, you see that even that response can classify non-survivor versus survivor, right? And then this is a sort of a motif emerging here, higher IgA response. We keep seeing it here. And then we also see that the pre-pandemic healthy actually corresponding to the compared to the eventually those who got SARS-CoV-2 infected have a higher antibody response against the endemic coronavirus as well. So that makes one sort of wonder, well, maybe having a higher response already to the endemic coronaviruses helps you in terms of survival or not getting that infection, right? Not clear yet. So that sort of brings me to the summary of this part of the talk. So we think this is a biomarker of survival, clearly uncovered by the multiplex antibody profiling pipeline that I described to you. This is from a day one sample, just to remind you, right? So when someone's going to the hospital, at that point this readout can be taken and potentially help in apportioning healthcare and so on. Canonical and non-canonical. Which canonical to which the response is well studied, non-canonical not so studied. Clearly they need to be studied is one of the conclusions here because the responses to them even can be predictive. But and also the endemic coronavirus responses are predictive as I showed you. And then the biggest surprise, as I said, that the pre-pandemic healthy actually have IgM response to the non-canonical SARS-CoV-2 antigens. So with this, in terms of the mechanism, we started thinking that maybe the switching of the existing IgM to the more functional IgA and IgG3 is involved mechanistically in being able to survive eventually the severe infection. So we think, in terms of a biomarker, definitely this is useful, but maybe this has also uncovered something where these antigens might need to go into future vaccines, right? If they mechanistically correlate in terms of helping out in severe, so maybe to prevent infection or development of severity at all, the current vaccines are maybe good enough, and that's what we're seeing even in the face of all these variants coming out. But post severity, maybe there are other antigens needed. So that's sort of showing the power of this pipeline or this workflow as well, where now it's a discovery engine, right? So it's not only biomarker, but new science as well, and maybe potential vaccine antigens and so on. So with that, so that's our preprint. I welcome you to look it up. So this we have preprinted already. Work remains in review. But in the rest of the time remaining here, I'll talk a little bit, since this is nano at tech, so I should go into the nano domain a little bit here. There's been too much biology, I guess, for some people. So I'll talk about this point-of-care diagnostics and transduction technique that we have developed, which goes into then can these multivariate biomarkers, these fancy things I talked about, right, 170 readouts, what use are they if they cannot be measured inexpensively? In the hospital or wherever, right? So ideally at the point of care. So eventually the current readout methods, all these lasers and photomultiplier tubes, they're great, very sensitive, very accurate, very expensive, right? So, and then the manpower, as I said. So the trained manpower required to run them comes at a cost as well. So these things make this, good to know for scientific research, great, but difficult to translate. So our thought has been, can we eventually take all of this and build it into a little chip somehow, right? That's the science fiction vision currently. That, take a drop of blood, put into that chip, and somehow on your phone comes a readout, right? That says, oh, you need to get better care because you are at risk of developing very severe COVID-19, for example, right? So this is this idea of portable and inexpensive point of care. So ELISAs are a kind of assay which are used to measure antibody, various immunobinding. So we want to develop something like this. So for this, we started looking at a lot of the cost. We looked at what adds to the in terms of the technology and the instrument cost. What adds most? Well, it is these lasers and photomultiplier tubes and expensive optics involved, for example, right? And then there's mechanics associated with them and so on. So we thought, and because I'm an electrical engineer, that's one of the bigger reason as well. We started thinking, can we bypass all of that, right? Can we go directly to an electronic readout? I mean, your phone has the electronic input. Let's generate directly an electronic readout somehow, right? So, we found this particular chemistry. So usually, it's a very similar chemistry to what is done, what I told you about on the beads and so on. But on the beads, it's a fluorescent readout eventually, right? So there's a fluorescent tag eventually on the secondary label. Here, it's an enzymatic tag which can do silver reduction, right? So you can see where I'm going here. The silver reduction can deposit metals, right? On the surface it's selectively bound to. This has been used for electron microscopy and so on, for example, because silver is sort of, it'll be visible in a particular way under an electron microscope being a metal. So that is where this has been mostly used. But we started thinking that this idea, which is called enzymatic metallization, which has existed, can we sort of try to build a direct electronic detection technique using this method here. So first of all, of course, we wanted to see whether we can even couple these to an antibody assay, right? So that's relatively simple. You coat whatever, some capture molecule, you put your samples on, put the enzyme probe on, and then the enzymatic substrate, it deposits the silver. That seems to work, right? There's just little wells on a glass slide. Healthy samples, not that much silver. COVID-positive samples, a lot of silver. You see a dilution curve, so that's just the sample being diluted, right? So you see the difference sort of, visually you start to see it already. But what about this electronic detection idea then? So, we thought, okay, so there's the silver deposition happening. We're going to put in some electrodes there. Some electronic property will change, conductivity, resistivity, whatever, and we'll have it. Well, not so fast. So we made the chips, of course, right here at IEN. It's just a very simple gold microelectrode array, so IDs, fingered electrodes. We put the chemistry on it. We see silver deposition, but it's sort of, you see how it is sort of getting repelled by the gold almost, right? So there's silver deposition happening, but it doesn't seem to cover the electrode somehow. So we were very, very curious what is going on here. And of course, what this ends up meaning is in terms of the electronic properties, it's not very repeatable or changing that much, right? So from there, looking into it in detail, we see that, so this is the chemistry I just described to you, but we start to see that yes, there is metallization, and then looking at it under the electron microscope reveals something very interesting. We see that there is the silver nanoparticles getting deposited, right? The enzyme coating was supposedly everywhere, but you clearly see that there's more silver on the electrodes versus the glass, right? And the glass is where we want it to be in order to bridge these electrodes. So that clearly is the reason it doesn't work somehow, right? And then you can zoom in and see that the gold has a very large amount of silver. It's almost a very dense silver on the gold electrode, but on the surface here, not that high. So of course, digging back into literature, we realize that of course, gold also can catalyze silver reduction. Is something that is known, and that's what might be happening here. It had been said. I mean, it's been said in literature. Well, gold has to be in a sort of a nano material or a nanoparticle format to be able to do that. Gold electrodes shouldn't be doing that, but they are doing it for us. So then we were sort of confused here. But we nevertheless said, okay, let's put in some gold nanoparticles in here. What will happen if we add some gold nanoparticles instead of the enzyme, right? So there's no enzyme, only the gold. It seems the gold does much better than the enzyme, so forget the enzyme, let's put in the gold, right? So now the tag is only gold nanoparticles. We see that we have metallization happening, but it's still pretty low and it doesn't conduct. In fact, it's lower than the enzyme case. So clearly the enzyme works better than the gold, and the gold does something in the nano format as well. But the best thing is the gold electrode somehow, right? It's generating all this silver, almost as if it's stealing the silver from where it needs to be, right? It takes on all of it, and there is not much getting deposited here. So with this, we came up with this idea that, well, what if there are gold nanoparticles and enzymes both there, right? What happens if you do that? And that was sort of the aha moment when we tried this. So what you're doing now, you have the nanoparticle probe, but you also have the enzyme probe, and suddenly you see a lot more, just visually, a lot more silver deposition, right? So this is not the addition of this and this, even visually you can see. It's a lot more. So some synergistic effect is happening here, which makes it a lot more than what the enzyme could do alone or the nanoparticles could do alone. They come together to do something different. And you then start seeing in the electron micrographs, there is a dense silver carpet everywhere, right? On the electrodes, it always had been, but now on the glass as well. Zooming in, you see a connected layer, a more connected layer forming, right, instead of these disconnected particles. So that told us that the nanostructuring of the surface by adding this gold nanoparticles clearly generates some kind of synergy with the enzyme, which we don't fully understand yet the mechanism of. And if one of you does, I'll definitely love to talk to you after. But this definitely seems like it could generate a signal for us, right? Because this is a looking continuous layer here. So we went in and measured, of course, the impedances. So this is just telling you that when you have only the enzyme, you have a high non-repeatability, as I said. It gets a somewhat conducting, right? I mean, the open circuit resistance of these things is above 100 mega ohm or something at this frequency. So you see that it's coming down a little, it's non-repeatable. If you have just the gold nanoparticles, as I showed, right, it's not conducting at all. There's some silver, but not conducting. But when you add these two things, it becomes fully conducting. It's almost a short circuit, basically, right? So from that 100 mega ohm down to like less than 10 ohms. So 6-7 orders of magnitude of impedance change if you do it this way. So this, we thought, hey, this is our electronic transduction technique, which can help us. We've seen that it can be coupled to an immunoassay. Now we have a way to turn it using the gold nanoparticles. Now we have a way to turn it into an electronic readout. So let's run with this, right? So that's exactly what we did. One question is this quantitative? Is this only yes, no, right? One would want a quantitative readout, of course. So what we've established here that yes, it's quantitative. So the way to read these plots, if you've never seen an ELISA plot before, this is the sample dilution, right? So highest concentration, lowest concentration. So 1 over this is the concentration kind of samples being diluted, right? And then you see that there is a quantitative readout happening. The blue versus red is basically just a matter of detail, 2 ways of adding the nanoparticle. You can add it after you have bound sample as a probe, right? Or you can even just decorate your surface already with the gold nanoparticle before even starting the assay. We see that decorating the surface ahead of starting the assay gives 2 orders of magnitude higher sensitivity, right? And this is just showing you the difference of the silver as it goes down, and these are the controls, which basically don't have much conductivity at all. So here, of course, you'll notice that I've flipped the scale here to make it look more like the ELISA curves that the biologists are used to seeing, which basically goes up instead of down. Of course, for us, the resistance actually goes down. But yeah, so here's the 10 ohm and the 100 mega ohm is down here. So you see a nice dilution curve happening. So that establishes that a quantitative electronic readout is actually possible. Of course, once you're microfabricating, this is just trivial, right? You can build a lot of these on a single chip. This is what we're talking about in terms of how computation became cheap and all of that good stuff. So we, of course, start to do that. This is a single microscope slide, 1 inch by 3 inches. Lots of electrodes on it. I believe it's 60 different. And you can see the visual readout is sort of there as well, right? High silver, low silver, COVID positive versus healthy. And we started calling it easy ELISA. It's an acronym. I can talk about it if you are interested. But the ELISA, the gold standard technique, what it sees in terms of the readout. So here's the optical readout, right, using the expensive instrument that I talked about. And you see that there's good concordance, definitely, but also much larger differentiation here happening, which leads to a slightly better sensitivity, specificity, this area under the ROC curve here, right? This is because of the switch type characteristic of this assay, right? Because it was not conducting, it became conducting. So a huge difference suddenly gets created. So that tells us that you can do high throughput electronic readouts using this. And then the last thing to be able to do those multivariate markers that I talked about, can this be multiplexed? Do I need to take many different samples in this case? So over there, there was the barcode and the bead and so on, laser and everything. What about here, right? So the question becomes, well, we know that it deposits silver where the sample and antigen had bound. How local is this, is the question we asked. Like if we had a small spot which had a positive sample, one antigen, and the right next was another antigen, would there be 2 independent readouts, or would they mix up, right? Because the silver deposition is happening from a liquid substrate. Does the reduced silver from one go to the next? Or how does that work out was the key question we were trying to ask. So we built a very simple reconfigurable, sort of, there are 4 electrodes here, very simple chip-wise. There's a resealable plastic, so PDMS layer, essentially, we're making wells. You pattern 4 antigens, 1 of them expected to be positive, 3 negative. And then you, of course, then want to put a single drop of sample, right? That's the idea. Can you do this measurement from a single drop of sample is what we want, right? So that's what we did. And then you see, wherever you put the positive mark, that's where the low resistance is, that's where the high silver is. And the others remain non-conducting, right? So 100 mega ohm versus less than 100 ohm. So that tells you that from a single drop of sample, by patterning different antigens, you can get a microarray type electronic readout here, right? So that was pretty cool. We wanted to apply it immediately to something interesting, and in this case, the easiest available sample at that constant. This was a little bit later now in the pandemic, right? Much more recent data. So we're like, vaccines are out, so let's look at how the response to a vaccine versus people who are COVID infected. Is it different? And that's expected to be different, as I've talked to you earlier, that the spike antigen is the only antigen in the vaccine. People who get infected end up developing response to many antigens. So we put in the spike and the nucleocapsid antigen here. The probe side also we multiplex here, so that's one of the beauties of this platform. You can measure IgGs and IgMs, right? So by putting different probes on the different wells. So that's what we did. And you see that, of course, the pre-pandemic healthies, they don't have, in this case, any response. So these are SARS-CoV-2 antigens only. So, of course, all that other stuff I talked about, the endemic viruses, is not relevant here. So they don't have a response at all. People who got infected have response to all the antigens. And in this case, it seems they still had an IgM response as well. So probably it was sampled early in their infection. And then the vaccinated people, this happens to be from my blood actually, but back then that is all we had access to. But you see that you have IgG spike antigen response, but no response to the nucleocapsid, which is not in the vaccine at all, and this tells me that I was probably not infected, right, till then. Because again, in this case, with this disease, you don't know whether you were infected unless you get, so this is my antibody test telling me that, oh, I only have a vaccine response. IgM is not here because this is 2 months after infection, so the IgM response is probably quenched by then, right? So with all of that, tying it together, you have these chips and electronic detection happening, multiplexing and so on. Of course, this is what finally enables it being pushed into translation, right? The idea is it's a simple electronic readout. You don't need, it's just resistance change. You don't need much to detect it, right? It can be done with a multimeter. A lot of data I showed you is with those handheld yellow multimeters that all of us have used in undergrad labs and so on. But we built a little portable LCR meter of our own as well, just to be a little bit fancy, I guess. So this is fairly simple electronics. There's a microcontroller in there, a single chip LCR meter, I think from Analog Devices in there, interfaced as well. And you can just run your assay with the drop of liquid on it, plug it in, and then this app one of the undergrads in the lab developed, which can show you a readout showing, oh, these two are positive, those two are negative, or generate a graph, or save the data to Google Drive, and all those good things it can do. But this basically now is getting to a point of care electronic antibody profiling. Of course, there's work to be done, but I think I've shown you that some of this is starting to become possible here. And so the next thing actually we are currently working on, which is more becoming more relevant now because the variants now don't lead to that much severe infections, right? But monitoring vaccine efficacy is a new need, right? Each of us would like to know, is my vaccine working, right? Oh, new variant came out. Is it working against the BA2, my vaccine? Is it still working, right? We want to know, but there is no such assay. I mean, yes, in the research labs there are assays, but they are so difficult to translate that most of us don't even know that these assays exist, right? So we're working on that, developing individual electronic vaccine efficacy assays. But looking beyond COVID, as I've shown you, wherever this antibody functional characteristics, glycans, and so on, can track with disease progression, you can apply the biomarker discovery and detection techniques. And we have some preliminary data showing in diseases like bacterial diseases like typhoid. So this is a viral disease I talked about, but bacterial diseases like typhoid, these markers can work fungal as well. Also, some completely non-infectious contexts, but antibody-driven rejection of grafts is a huge problem with people who get transplants. So we think we can be able to predict earlier whether a rejection is occurring by measuring these antibodies and glycans. And of course, eventually, why just antibodies, why just blood, right? Because it's a simple immunobinding assay that does not really depend on these specifics. So we think, we can be more ambitious and see, oh, can we compete with PCR? Right? Can we be so sensitive, right? PCR has a 2 to the 40 amplification, right? 40 cycles. So that's a large number. Can we be so sensitive to displace PCR as well now? That's a tall order. We are far from there, but that's something we want to try. And of course, then larger objects. I think this should actually be more trivial and easy, but we haven't yet done it. Cell-based assays, right? Bigger objects. Something binds, you can detect it, is the idea. So why not cell-based assays, right? Cell counts, immunophenotyping, functional assays, all of that. So I think I'm running out of my time here for sure. So I'll just acknowledge the team, some of whom are here in the lab, small but growing team. And of course, none of this can happen without the clinical collaborators who provide the samples and the understanding of immunology and so on. And of course, acknowledging my funding sources here. So with that, I'll take any questions you might have.
Questions from anybody here in the room?
Quick question. More on the microfluidic part. Yeah. So the question on the microfluidics part. So obviously there are probably mixing constraints on the time response, yes. And maybe even there are some, I don't know, because maybe some crossover between different electrodes that can influence the response. Can you comment on those kind of engineering challenges in these types of technologies? How important these are and what to pay attention?
So I think, awesome question, and that's something we worry about a lot. We've been lucky a little bit that the crossover effect is not so much there, but mixing, we think, in terms of improving the repeatability of some of these assays, especially down at where the very low, picomolar and so on, we think that could start mattering. We would like to do it. We haven't currently done it, right? Do we want a microfluidic mixer separately, or some electrokinetic mixer, or what have you? We don't know yet. So we'd like to go there, haven't gone there yet.
Okay, great.
Hi, great talk, I enjoyed it. I had a question about something you mentioned earlier. You said that the impedance measured at that particular frequency was satisfactory, but I realized that maybe you're running things at different frequencies.
Yeah.
So have you found that there's some
Yeah.
paths to explore, like an impedance spectroscopy type of approach, or just curious what you
Right,
found on this
right.
spectrum?
So that, if I tell you too much, I'll give away some of the results, newer ones, which my students may not like. but definitely, no, I'm just kidding, actually. So the thing is, this is a resistor eventually at the two ends of it for sure, right? So we did a whole spectrum with the LCR meter, and that's why the portable thing is an LCR meter, because back then we didn't know what will happen, so we started developing a portable LCR. Turned out that it's mostly a resistor, right? So we see a clear capacitive, the kind of thing you would expect a spectrum versus a bare resistive spectrum. Now, but does that mean that it can't be converted into something that will be more sensitive if you do a full spectrum? That's watch this space type of thing. It's coming, yeah.
Hi, Aniruddh.
Hi.
Excellent presentation. Thank you. One of the questions I have is that, in your sandwich assay, what is your fluorescein tag? And number two is that, did you observe any photo bleaching associated with when you use that laser?
Yeah, so I think, in these, so maybe you or others here are aware of the Luminex platform. That's what this is. The classification lasers, I think, are rhodamine and some IR dye. And those are pretty high amounts inside the bead, so it doesn't matter much. The back-end probe readout is just phycoerythrin. That it does matter, so one has to be careful to not expose it too much to ambient light and so on. But eventually, within the limits of differences that we want to look at, it seemed to be fine, is what I'm able to say. Yeah.
Thank you for your talk. A couple of questions. The first one is, how do you immobilize your antigens onto the glass part of your chip? The second one is, you've shown some results of sensitivity of your device for different dilutions of your sample. How does it translate into actual concentration of antibodies, and what is the limit of detection? And the last question I had for you is, okay, once you put a drop of sample onto your device, I understand that there's different steps of washing, incubating with other samples and stuff. How long does it take actually the entire process? Thank you.
Yeah, those are all great questions, and there's a lot of work, I would say, remaining to be done in some of them. The immobilization question, let me get it out of the way. We use this molecular adhesion layer standard one called polyl-lysine. It's a positively charged, so it's a charge-based thing. So that just works for us at least. In terms of the sensitivities, so now with clinical samples, it's a little bit difficult, to define how much exactly antibody they had in terms of nanograms per. It can be done by running a calibration assay, right? For example, like a separate ELISA with a standard or something. So with the clinical samples, I don't know. I do know that our sensitivity currently, and this is based on data I have not shown here, is somewhere in the middle of what the ELISA instruments can do versus what your lateral flow assays, which just do a yes/no. So we are pitched somewhere in the middle there. We'd like to go towards the full ELISA, right? But we are not there yet, I think. And eventually the point, I think, becomes we get lucky here because antibodies are such in large amount in the clinical serum that we are able to work this out. So the bead-based assay I talked about, right, for the immunology and the biomarker detection aspect, that's a sandwich assay. That's not even enzyme amplified, and it still works, right? Of course, that's with the laser. So what that tells you is that this is, for this application, seems good enough, but yeah, we need to find out what the exact limits are and improve it. And then there was a third thing you asked. Oh, yeah, sure. Right. So that's where the microfluidics comes in, and we'd like to automate it. Currently it's long, let me just be frank, because the incubation with the sample itself can be half an hour or an hour, and then there's washes and so on, basically, yeah.
Okay, I think we are at the top of the hour, so I'm going to thank Aniruddh one more time, and thank you for coming.